This is a production of Cornell University.
Hear me? Thanks, Lynn, for that wonderful introduction and for decades of friendship and professional collaboration. And thank you for the opportunity to share our research program at the Danforth Center with Cornell and everyone who's doing some fantastic plant science here. You might see some familiar faces, some familiar references to Cornell folks. As Lynn said, I'm the director of the X-ray imaging facility at the Danforth Center. And if you are not already aware, the Danforth Center is the world's largest nonprofit independent plant science research institute. And we have the fortunate position to really look into a lot of issues in plant science, especially food security, that other places don't really have the freedom or the support to do. I'm a research scientist in Dr. Chris Topp's lab. Chris is the PI, and he started the lab in 2013 using phenotyping to look at root biology, and that has expanded to include not only roots and genetics, but everything surrounding the roots. So there's a wide range of expertise that we share. And as I expect many of you here are starting to realize that no one lab will have nearly enough expertise to get everything done. You must collaborate, you must find ways to work with your colleagues to really pull more of the science out of all these projects. So you can see the list on the left: plant genotypes and genetics and phenotyping, my background in microscopy and imaging, there's cell biology, there's ag engineering, chemical engineering, and perhaps most importantly is computational science, because all the beautiful images are mostly just greeting cards until we can actually pull some of that biological information back out the other end of these really cool 3D volumes. So a couple different headings today. This is lab-based X-ray microscopy as opposed to working at a synchrotron. The lab-based makes it very, very accessible. There's, of course, trade-offs compared to synchrotron imaging. So I'll cover X-ray tomography. Folks often say it's micro CT or nano CT or macro. No, that's just the resolution you're dealing with. It's all the same instrument. So we have an X-ray tomography instrument. We have an X-ray microscope, which generates images in a slightly different way. There's specific application in volume electron microscopy. That is using XRM as a roadmap to show you where in your resin block you want to go for organelle level volume imaging with a serial block face SEM or a FibSem, other the instrumentation that does tomography at the nanometer scale. We've done some really sort of exciting pioneering work looking at mycorrhizal fungi in situ, which is a really big challenge. And we've been recently getting into looking at X-ray imaging of field soil to try to see what can we understand about the 3D structure of soil at multiple scales. So to begin with, coming up on 8 years this August, we took delivery of a large format industrial scale X-ray tomography instrument. This has been in collaboration with Valent Biosciences, who make a lot of biorationals and mycorrhizal products for the commercial market, in addition to part of an NSF grant. Now, these large-scale industrial instruments are frequently found in automotive and aerospace. Boeing will probably have a dozen of these to look at aircraft parts. They have to do non-destructive imaging of aircraft parts to confirm that they're in good shape. And having taken 2 different flights to get here, I'm glad they do that. They know the parts are working, but they have to certify that all the parts are good. So they have very large cabinets. They can handle very heavy weights. They have a lot of interior space to work with. And these were the advantages that we found in the large format instrument. A quick review of how we generate image data using X-ray tomography. The setup is relatively straightforward. You have an X-ray source. Sample sits on a turntable in the middle, and you have a detector which collects the image. We broadcast X-rays through the sample that impinges on the detector. We'll rotate that turntable over typically 360 degrees. We'll collect thousands of images from around all those angles. and then computationally reassemble all those images into a 3D volume that we can now explore electronically. A lot of the animations that you'll see today, certainly the colorful ones were done by my senior technician, Clara Lebo. She's been developing her undergraduate degree in art, her master's degree in electron microscopy, and combining them to really make some stunning and striking ways of presenting these 3D volumes. The practical offshoot is we can study these things without tearing them apart. If you wanted to count every seed inside this pepper, you could do that digitally without having to hack it apart. Some of the very first work we did was actually in collaboration with Jason Londo, who's at Geneva. Hopefully you're online there, Jason. Hello. They provided hundreds of grape rachises. These are the stems left behind after the berries of the grape are gone. And if you want to improve grape varieties, you can't just focus on making big berries. You need to understand the underlying 3D architecture of what's going to hold those berries and the enormous amount of variability that already exists in these systems. So certainly with our eyes, we can look at all the different grape bunches and see that they're very, very different, but how do we quantify these differences? So what we did is we scanned hundreds of these grape rachises, and when you are fortunate enough to work with an applied mathematician like Mao Li, whose one of her expertise areas is looking at topology. How do you evaluate a 3D structure? And so she uses something called persistent homology, which takes a geodesic distance that you measure from one end of a complicated structure to the other, and the timing at which various components fuse into one another or disappear can be mapped as a barcode. And this animation illustrates as you go from the tips down into the central trunk and down, the various components start to disappear and get melded into one. And these can be portrayed as a barcode. And so what that allows you to do is take many different complicated branching structures and mathematically distinguish them using this barcode sort of architecture. Again, this is Mao's expertise, certainly not mine. So I am exceedingly fortunate to work with such experts. Margaret knows Mao very well also.
Quick question.
Yes.
The bell pepper that you had up there before, assuming that the instrument and everything is all set up, how long does it take for the machine to do the scanning to generate the information which then ultimately leads to the video?
Yeah, so the question is how long, specifically that bell pepper scan, how long does it take to image? And for that particular bell pepper, it was about 30 minutes. It's relatively non-dense, relatively easy imaging. So a 30-minute scan, typically 3,600, 4,500 projections. We can do it shorter. It will be lower resolution. We can go longer, higher resolution. So there's a window within which we work. But for something low density like that, 30-minute scan covered that. So jumping it back into persistent homology, it's a way to measure all these unique 3D scans of all these hundreds of grape rachises that we did. So I provided all the 3D data to Mao. She ran these 3D shapes. We extracted surface volumes. She put the surface volumes through a persistent homology measurement tool that she engineered, and then used linear discriminant analysis to group these persistent homology measurements based on their characteristics. And what turned out was this LDA grouping of all the phenotypes, all the different 3D measurements of the grape rachises, corresponded very nicely to the actual genotypes. And it mapped very, very well. So the very broad range of shapes actually linked very nicely to the differences in the genotype. So Mao was really the spearhead of the analysis side of that. Again, I had the easy part. I just kept putting rachises in and scanning them and getting these wonderful data back from Mao showing what we were learning from it. But the other thing we wanted to learn was, can we use this in a predictive sense? Can we scan actual grape bunches and start to incorporate the size of the berry and the distribution of the berries along the grape rachis to start to come up with modeling rules for how does a bunch of grapes form? What are the rules that govern the shape? Does it stop at a certain point? Does it stop when it contacts? Does it stop at a certain weight? And these are some of the measurement data that went into this paper that that Mao again wrote, and Jason was part of, I was a part of, Dan Chitwood, a number of folks who really contributed to understanding more about how these objects occupy 3D space. And so again, this is just an animation that Mao put together that illustrates using these rules how berries might grow and how they form, but it begins with mapping the rachis itself and then adding the modeling component that we got from the berries. Sorghum panicles. Sorghum is a very important crop for food security in many areas of the world. It grows often in places where corn does not. From the image here, you can see that it's tremendous variability in panicle architecture. When I started at the Danforth, there were a few scientists who had collections of hundreds and hundreds of different panicles from an association panel. And so we realized that an X-ray scan of an entire panicle would give us a really data-rich information to work with. So not only could we see every individual grain, but then we could also see the underlying architecture of the panicle itself, all the branches that hold on to the grains. So again, if you're breeding sorghum and you want more grains, you need to understand what the impact is on the supporting architecture. And again, we're very fortunate to work with very talented postdocs. Mao, again, was part of this, but Ray Schau, who's now a scientist at Wageningen in the Netherlands, they took all the data. We scanned over 900 sorghum panicles. And what they were able to do was to use these scan data to, as you can see from this graph, map where along the panicle the most grains were deposited. Was it most in the middle, most at the ends? And how did that vary across the 900 in the population? Zoom in and actually analyze the shape of every individual grain. How round were they? How spherical? How ellipsoid? And how was that shape range distributed across the 900 varieties? And then electronically remove every single grain, not by hand, but electronically, and then leave behind the branching architecture and map that. So now these are all mappable, measurable 3D trait data, phenotype data that can be compared using multivariate statistics amongst all these 900 different varieties. And what they found, that the conventional wisdom was the shape of the panicle dictated the genetics. And in fact, that was not the case. You could find the same examples of panicle architecture across all the genotypes. So that was not the measure of interest. And again, working with all-stars like Toby Kellogg in the field of grass and grass biology is just one of the benefits of being at the Danforth, surrounded by just really phenomenal people that raise your game. Far more important than that is a distillery located in downtown St. Louis, where a friend of mine, Dave Weglertz, makes a really spectacular sorghum whiskey. So if you ever get to St. Louis, you have to go to this distillery. It's just 4 blocks from Busch Stadium if you go to the ball game. And he'd love to give you samples of the sorghum whiskey. And you can see off to the side some of the sorghum panicles. So always some practical input from the imaging that we're doing. Again, I told you the top lab started out looking at corn roots and corn root biology. So how does one actually measure and phenotype maize roots when obviously everything important is happening below ground? So we've been breeding plants as humans for thousands of years, but only breeding what we can see and improve. And along with that came root traits. They are there, they vary, but we've not been able to work with them very efficiently. So when you look at these 15 different maize root crowns, it's a very informative region, right where the roots come together, the nexus, right at the soil line. They were all grown in the greenhouse in an artificial medium in great big tree pots. So the environmental conditions were as identical as we can practically make them. So you see the huge variety evident. Clearly the genes are playing a big role in making roots decide how to occupy 3D space. We want to get a handle on those genes. We want to find a good strategy for doing that. So for us, it begins with measurement. So we'll plant breeding populations out in our research fields, and then the whole lab goes out there with shovels and lots of coolers of water and all sorts of bribes. And it's literally shovelomics. A shovel will dig up a useful volume right around that root crown. And then they all get brought back to the lab. They get washed. We have washing stations and dried. And that gives us really a very rich data source that describes 3D architecture because the maize root crowns are relatively well lignified at this stage. They hold their shape pretty well. So comparisons between genotypes are valid.